Not all pathogens attack from the outside. Some are far more deceptive, slipping silently inside our cells and turning them into hidden shelters. Once inside, these intracellular pathogens become completely invisible to antibodies, placing them beyond the reach of the humoral immune response. This is where cell-mediated immunity steps in. Specially adapted to detect what lies beneath the surface, this arm of the immune system patrols the body for infected, stressed, or abnormal cells, identifying subtle molecular signals that reveal the presence of an internal invader. Rather than targeting free pathogens, it targets the compromised cells themselves, marking them for destruction and stopping the infection at its source. In this battle, precision is everything, and cell-mediated immunity provides the tools needed to fight enemies hiding from within. The immune response now reaches a decisive checkpoint, the activation of a naive CD4 T cell. A dendritic cell, carrying peptide fragments of the invading pathogen, approaches as a highly specialized messenger. Activation, however, is not accidental. It requires three tightly regulated signals. First comes signal 1. The T cell receptor on the CD4 T cell precisely recognizes the antigen displayed on the dendritic cell's MHC class II molecule. This interaction provides specificity, ensuring that only T cells with the correct receptor are engaged. Next is signal 2, the co-stimulatory conformation. B7 molecules on the dendritic cell bind to CD28 on the T cell, delivering a critical message. This antigen represents real danger. Without this signal, the T cell would remain anergic or functionally unresponsive. Finally, signal 3 delivers direction. Cytokines released by the dendritic cell, most notably interleukin-12, shape the fate of the activated T cell, guiding it to differentiate into a T helper 1 effector cell. Through this final signal, the immune system determines not just whether to respond, but how to respond. Together, these three signals act as a stringent security system. Only when all are present does the CD4 T cell fully activate, ensuring a powerful yet precisely controlled cell-mediated immune response. Cross-priming is illustrated as a clever tactical move on the immune battlefield, where a dendritic cell patrols the tissue and collects fragments from virus-infected or tumor cells, even though it is not itself infected. Instead of following the usual rules and presenting these external antigens on MHC class II, the dendritic cell reroutes them into the MHC class I pathway, displaying the foreign peptide like a warning signal meant for CD8 T cells. A naive CD8 T cell recognizes the peptide MHC1 complex and pauses, awaiting confirmation, until a nearby CD4 helper T cell delivers a licensing signal through CD40-CD40 ligand interaction. With all signals secured, the CD8 T cell transforms into a cytotoxic T lymphocyte, ready to hunt infected or malignant cells, illustrating how cross-priming prevents pathogens and tumors from escaping immune detection. Once fully activated, the CD4 T cell undergoes a critical transformation. Shaped by its cytokine environment, it differentiates into a T helper 1 cell, a master coordinator of cell-mediated immunity. In this form, the T helper 1 cell emerges as a powerful commander, radiating key cytokines such as interferon gamma and interleukin-2. These molecular signals act as strategic orders, mobilizing immune forces specialized in eliminating intracellular threats. Interferon gamma drives the activation of macrophages, sharpening their ability to destroy pathogens hidden within cells, while interleukin-2 fuels the expansion and differentiation of CD8 cytotoxic T cells. Through this central leadership role, T helper 1 cells do not fight alone. Instead, they orchestrate a coordinated and highly effective immune response, ensuring that intracellular pathogens are detected, targeted, and eliminated with precision. The command signals released by T helper 1 cells now take effect. Interferon gamma, a defining T helper 1 cytokine, binds to nearby macrophages and triggers a powerful transformation. Macrophages engulf the invading pathogen, trapping it within their phagolysosome. But the macrophage does more than destroy. As an antigen-presenting cell, it carefully processes the pathogen, breaking it down into recognizable fragments. These antigenic peptides are then displayed on the macrophage's surface, mounted on MHC class II molecules like warning flags. Once activated by interferon gamma secreted by T helper 1 cells, macrophages transform into the M1 type, the classically activated, highly aggressive form specialized for intracellular killing. Their internal machinery intensifies, lysosomes multiply, phagolysosome fusion becomes more efficient, and microbicidal mechanisms are fully unleashed. What were once simple sentinels are now elite executioners, capable of destroying the pathogens they have engulfed. This moment highlights a critical feedback loop in cell-mediated immunity, where activated T cells empower innate immune cells, amplifying the body's ability to eliminate hidden intracellular threats. The T helper 1 commander now turns its attention to building the strike force. By releasing interleukin-2, the most critical growth factor for T cells, naive CD8 T cells are activated, driven to proliferate, and guided toward full differentiation. One cell becomes many, and many become specialized. As this clonal expansion unfolds, the CD8 T cells mature into cytotoxic T lymphocytes, forming a powerful army trained to recognize and destroy infected or abnormal cells. Interleukin-2 ensures that this response is not weak or isolated but large-scale and effective, providing the numbers needed for a decisive cell-mediated immune attack. Attention now shifts to the infected cell itself. The intracellular pathogens' proteins are cleaved into peptide fragments, which are displayed on the cell surface by MHC class I molecules. Unlike MHC class II, which presents antigens from outside the cell, MHC class I reveals what is happening within. A passing CD8 cytotoxic T lymphocyte scans these molecular displays and instantly recognizes the non-self peptide bound to MHC class I. This precise interaction signals that the cell has been compromised and must be eliminated. The specificity of this recognition ensures that only infected or abnormal cells are targeted, preserving surrounding healthy tissue while stopping the infection at its source. With the target confirmed, the cytotoxic T lymphocyte moves in for the kill. Locking onto the infected cell, it releases its lethal arsenal. Perforin is deployed first, punching precise pores into the target cell's membrane, creating an entry point. Through these openings, granzymes are delivered directly into the cell, where they trigger a powerful cascade of caspase activation. Within moments, the infected cell initiates programmed cell death, or apoptosis. This controlled destruction ensures the virus is eliminated without spilling inflammatory contents, demonstrating the precision and efficiency of cytotoxic T-cell-mediated killing. In some cases, cytotoxic T lymphocytes employ a different, equally lethal strategy. On their surface, they express FOS ligand, which binds to the FOS receptor on the target cell. This interaction acts as a molecular death signal. Once FOS is engaged, an intracellular signaling cascade is triggered within the target cell, leading to activation of caspases and initiation of apoptosis. There is no membrane rupture, no external damage, only a precise internal shutdown. As the cell succumbs, the message is clear: caspases activated, shutting down. Unlike cytotoxic T lymphocytes, natural killer cells are part of the innate immune response and require no prior sensitization or antigen-specific training. Instead, they patrol the body searching for a dangerous clue: the absence of MHC class I. Many viruses and tumor cells attempt to evade immune detection by down-regulating MHC class I expression, but this strategy only exposes them to natural killer cell surveillance. Detecting this missing self-signal, the NK cell acts immediately. With this rapid, non-specific response, natural killer cells provide early and essential protection against virus-infected and malignant cells. The immune response now showcases a powerful collaboration known as antibody-dependent cell-mediated cytotoxicity, or ADCC. In this scenario, immunoglobulin G from the humoral immune system first marks the target, often a tumor or infected cell. Natural killer cells then step in, recognizing these antibodies through their CD16 receptors, which bind to the exposed FC region. Once engaged, the NK cell releases its cytotoxic granules, triggering apoptosis of the antibody-coated target cell. ADCC highlights the remarkable teamwork between antibodies and cellular effectors, demonstrating how different branches of the immune system unite to eliminate dangerous cells efficiently and precisely. Once the intracellular pathogen is cleared, the battle is over, and balance is restored. The tissue is once again clean and healthy, but the immune system does not fully stand down. Remaining behind are memory T cells, silent sentinels positioned to respond if the same threat ever returns. These cells carry the molecular memory of the encounter, allowing the immune system to react faster, stronger, and more effectively upon re-exposure. Fast, precise, relentless, cell-mediated immunity protects from within. The story now takes sobering clinical turns. What happens if our cell-mediated immunity is compromised or goes berserk? Let me give some examples. Human immunodeficiency virus, or HIV, strikes at the very heart of cell-mediated immunity by specifically targeting CD4 T cells, including T helper 1 cells, the master coordinators of this response. As these cells are progressively destroyed, the immune system begins to shut down. Without T helper 1 cells, macrophages are no longer effectively activated, CD8 cytotoxic T cells lose critical support, and coordinated cellular defense collapses into chaos. The consequences are devastating. Intracellular pathogens and malignancies that were once controlled now flourish. This profound breakdown of cell-mediated immunity explains the hallmark of acquired immunodeficiency syndrome, or AIDS, severe opportunistic infections emerging when CD4 T cell numbers fall. Another tragic consequence of immune dysregulation is autoimmunity. In this scene, the immune system misidentifies the self as non-self. A CD8 cytotoxic T lymphocyte targets a pancreatic beta cell, a vital cell that produces insulin. Displaying self-antigens on MHC class I, the beta cell is mistakenly recognized as a threat and marked for destruction. This process illustrates type 1 diabetes mellitus, a disease driven by T-cell-mediated immunity rather than antibodies. This highlights how the same powerful mechanisms that protect the body can, when misdirected, cause profound harm.